At Stealth BioTherapeutics, we are focused on mitochondrial medicine. We have first-in-class novel agents that we are using to treat genetic, inherited mitochondrial diseases, as well as a number of common diseases often associated with aging, in which mitochondrial dysfunction is a contributory factor. The mechanism of our agents is that they normalize mitochondrial function in dysfunctional mitochondria. We see no effect on normal mitochondria, and we do see end organ benefit across a number of different disease models. So mitochondria exist in almost every cell in our body. They produce about 90% of the energy that we need to survive, and they are important contributors to many cellular level functions. But when they become dysfunctional, and this happens with genetic mutations that are inherited, it happens as part of the normal aging process with disease and with injury and with environmental factors, they start to contribute to cellular senescence and to chronic inflammatory cascades. So again, when we are young, our mitochondria are producing adequate amounts of ATP to fuel our major organ systems, highly energy-dependent organ systems such as the eye. But with aging, spontaneous mitochondrial DNA mutations and accumulation of free radicals leads to mitochondrial dysfunction. And at that point, the mitochondria start to spew out additional free radicals, leading to end organ damage. What we see with elimipritide, which is our first-in-class mitochondrial protective agent, is that it diffuses across the cellular and outer mitochondrial membranes. It binds in the inner membrane of the mitochondria with cardiolipin, a phospholipid found only in the inner mitochondrial membrane, which plays an important structural and functional role in the oxidative phosphorylation process by which our mitochondria generate ATP. Cardiolipin is susceptible to degradation by reactive oxygen species, and so in a disease state, with the cardiolipin peroxidized, the mitochondria becomes dysfunctional with decreased ATP production and increased ROS generation. We see across various preclinical models that elimipritide stabilizes cardiolipin in the presence of increased ROS. So we see increased ATP, decreased ROS, normalized cardiolipin content, decreased inflammation, fibrosis, and cell death, and we see morphological remodeling of the mitochondria. So this is these images are electron microscopic images of mitochondria. On your left, you see a normal mitochondria, an elongated shaped organelle, dark gray, indicative of good inner mitochondrial membrane content. In the middle, with disease, this is ischemia reperfusion, the organelle becomes fat and bloated and loses much of its inner membrane. With elimipritide, we can protect against this, and we can also reverse the damage, and this is in a diabetic retinopathy model. We have a robust pipeline, but from an ophthalmic perspective, we are focused on dry AMD, Leber's hereditary optic neuropathy, and Fuch's corneal dystrophy. You see here that mitochondrial dysfunction is a hallmark of many ophthalmic diseases. And we know that in dry AMD donor eyes, mitochondrial dysfunction precedes vision loss and increases commensurate with it. We know from preclinical models that we can deliver drugs via topical ophthalmic drops to the back of the eye on your left and systemic delivery on your right. But these are not always predictive of what we will see in human eyes. In our first clinical study, phase 1-2 study, in 15 patients with dry AMD, 15 patients with DME, and 6 patients with dry AMD, elimipritide was found to be safe and well tolerated. These were topical ophthalmic drops, and reduced central subfield thickness in 2 out of 15 DME patients with no change or worsening vision in the fellow eye. But this is not a small study and potential biological signal. In Scott Cousins' model of dry AMD in a mouse model, After 1 month of systemic administration of elamiprotide, he saw reduced oxidative stress, improved cellular integrity, reversal of drusen deposits, improvement of membrane potential, and improvement of B wave, indicative of visual improvement. We are conducting a phase 1 open-label study of 20 patients with high-risk drusen and 20 patients with non-central GA. This is with 6 months of once-daily subcutaneous elamiprotide administration, looking for improvement in low luminescence visual acuity, in this case compared to natural history because we have no placebo arm, looking for improvement in vision speed, as well as improvement in drusen volume. We are also conducting a study in Leber's hereditary optic neuropathy. This is fully enrolled with 12 patients receiving topical ophthalmic drops twice daily for 1 year. We expect data early next year from both of these studies. And our final study in Fuch's corneal endothelial dystrophy, a front of eye disease, we are escalating dose to try our 3% ophthalmic drops for the first time in humans. These were more quickly efficacious in animals, and we hope to see benefit with these drops in this study. Data expected first half of next year. So we hope that with this novel treatment restoring normal bioenergetics to the mitochondria, that we can be of benefit across many diseases, including a number of ophthalmic diseases. Thank you so much.